So this week we're going to be talking about the endospore stain. So the endospore stain is another type of differential stain. So it involves the use of two dyes to show us two different parts of a cell. So what it's going to show us are vegetative cells, which are active cells, and then endospores, which some bacterial cells can produce when they're in a harsh environment to protect their genetic information. So it's kind of a stage of dormancy for a bacterial cell. So typically, not all bacterial cells can produce endospores, but typically they will produce endospores when they are subjected to a harsh environment. So they're never going to produce an endospore if the environment's stable. The environment has to become unstable, which would mean maybe there isn't enough food, the temperature is too high or too low, maybe there's some type of toxic chemical, something that would create an unfavorable environment for the bacteria would cause it to go into sporulation and produce an endospore. So here we have just an active, regular vegetative cell. It's in a good environment, it's growing, it's dividing, so it's stable. If it would get put into a harsh environment, and it's able to, it would induce sporulation, which would mean it would produce an endospore. Basically, the endospore is a protein covering that surrounds the bacterial cell's genetic information and protects its genetic information. So here's the rest of the vegetative cell, what's left of it, and then there's an endospore in here protecting that bacterial cell's genetic information in that harsh environment. Eventually, as more time passes, the rest of the vegetative cell will kind of shrivel off and die off, and the only thing that will be left is this endospore. And that endospore can sit for a variable period of time. I mean, it could be hours, it could be days, weeks, months, years, depending on the species of bacteria. It could sit there in that harsh environment, protecting that bacterial cell's genetic material. If the environment were to stabilize and those nutrients would come back and the temperature would be normal again and the toxic chemicals go away, this endospore would burst and it would be the return of the active vegetative cell again. And then if that vegetative cell feels threatened, it can again produce another endospore, so it can kind of cycle through this. The reason why that endospore is able to protect the bacterial cell's genetic material is because it does have a tough protein layer surrounding it, and that protein layer is actually made up of keratin. That's what makes it so tough and durable. And if you remember, it's the same keratin that's in our skin that makes our skin tough and durable. So that keratin really does serve to protect that bacterial cell's genetic DNA in that harsh condition. So typically when a bacterial cell produces an endospore, the endospore is inside the vegetative cell originally, and then again, as more time goes by, the vegetative cell completely shrivels up and dies off, and the endospore is the only thing that's present in that harsh condition. So the endospore initially, when it's in the cell, can be present in various locations. Sometimes it's just in the middle, which we call central. Sometimes it's between the middle and end, which we call subterminal, and then sometimes it's all the way to the end, which we call terminal. Now again, this is when it's inside of the cell. As more time goes by, that cell shrivels up and the endospore is just kind of outside of the cell. We can also look at the actual shape of the endospore. Over here, we're looking at more elliptical endospores. These are all more oval in shape. But there are spherical endospores that are just perfect little circles as well. So with the endospore stain, like I said, it is a differential stain. So it's done to look at vegetative cells, which are the active cells, and then the endospores, which are kind of the dormant phase where the DNA is being protected. Again, not all bacteria are able to produce endospores. Oftentimes, it's just your gram-positive rod-shaped or bacillus bacteria that are able to produce endospores. Not even all of them can. So typically, bacteria within the Bacillus genus or the Clostridium genus are known to be endospore producers. There are definitely some very dangerous bacteria that can produce endospores, but then there's also some non-pathogenic bacteria that can produce endospores too. So just because it produces endospores doesn't mean it's like a life-threatening bacteria. But from this list, we can see the bacteria that causes things like anthrax and tetanus and botulism, all fairly dangerous infections, are able to produce endospores. That means basically when these bacteria are subjected to harsh environments, they are able to protect themselves by producing an endospore to protect their genetic information in that harsh environment. And then once that environment stabilizes, they can become active vegetative cells again. So their survivability is a little bit better than what we would expect for just a normal bacterial cell that cannot produce an endospore. So how it works is you initially do all your smear prep that we've went over before, as far as putting the drop of water on the microscope slide and then adding the bacteria and then air drying it and then heat fixing it and then letting it cool down, and then you would go through the actual staining procedure. So the first stain that we use in a endospore stain is called malachite green, and typically when we use malachite green, we do subject the slide to heat through steam as well. That just kind of helps break down the keratin in the endospore so that the endospore can actually absorb the stain. So initially, when we stain with malachite green in heat, all cells are going to stain green, whether it's an active vegetative cell or a cell that's actually produced an endospore. Initially, just everything stains green. The next step is that differential step. It's what's going to separate the vegetative cell from the endospore. And that is just a simple rinse with water. So you would rinse with water, and that's going to pull green out of anything that's not an endospore. So this entire vegetative cell that has not produced an endospore, probably because it's not in a harsh environment, is going to be clear at this point. And this vegetative cell over here that has an endospore within it, the actual cell is colorless, but the endospore is going to stay green. The very last step is to counterstain with safranin. So safranin is going to stain any vegetative cell pink. So over here, this entire cell is going to be pink. It's endospore negative because there's no endospore within. And this cell over here, we see a pink cell, but it has a green endospore inside. So this cell over here is endospore positive. And like I said, the more time that goes by, the rest of this cell will die off and the endospore will be the only thing that's present. So this is what this would look like in a real setting. I mean, we definitely see like a few actual just whole vegetative cells that did not produce any endospores, probably because they didn't feel threatened or maybe they're not able to produce endospores. But we can definitely see some vegetative cells over here that have produced endospores. We see the endospores within, so we know these bacteria are definitely in a harsh environment and they're kind of in survival mode. And again, the more time that goes by, the less we'll see of this pink and just start to see the green. So there is a video in D2L that I want you to actually watch to look at the procedure, because oftentimes when I kind of explain it, it's difficult to imagine. But you would start off with just your general smear prep to prepare your slide. Then you would need to add that malachite green stain. We add that for 5 minutes, and we typically do this when the slide is exposed to steam. Again, that just helps break down the endospore so it can actually absorb the malachite green stain. After the 5 minutes with malachite green under heat, we rinse with water, and then you counterstain with safranin for 1 minute and rinse with water. And then all you have to do is blot it dry and then look at it under the microscope under the oil immersion lens. Typically your active vegetative cells would be pink, and then the endospores should be green. But do watch that video in detail again so you can familiarize yourself with the process. And then I also want you to use that bacterial staining virtual lab PowerPoint in D2L to practice an endospore stain. So just to review what that looks like again, you would go to your bacterial staining virtual lab and you would download it and open it. And then enable editing, and then click on this little slideshow button down here, and then go ahead and hit next, index, And then we need to pick our endospore stain, and then just read through the few descriptions they have, look at the procedure, and then just follow the instructions and click next through here as you actually go through and perform an endospore stain.